And also, let's try to look at AlphaFold's predictions for protein-protein complexes, because there have been some suggestions in the literature that it does very well, and maybe we should just use it to see how proteins bind to each other. And here's what I'll show today. So this talk is looking at protein structure from a very didactic point of view for about 5 minutes. And the subtitle would be, let's actually visualize what the problem is, rather than just talking about it. So that's what I want to show here. All right, so to start what we want to talk about, I want to talk about how do we visualize protein structures? How can we see if AlphaFold is right or wrong? And so a little bit of tutorial, and then we'll look at a bunch of AlphaFold runs for protein complexes. And so the title is 9 Views of an Alpha Helix, or Let's Actually Visualize the Problem. So here's the outline, some motivation. How do we actually visualize protein structures? Not as points, but as, some object. Let's look at how to visualize what's an alpha helix, what's a beta strand, how does AlphaFold do, and then look at AlphaFold on proteins binding each other. And I'll do a little demo with PyMOL at the end. I actually have done an analysis, and there's some room for hope. And the first part is mostly for Cynthia, because what is protein structure and how do we visualize it? So we're not confused. But I think you'll all enjoy it. Okay, so just to motivate this, the basic idea is, can we use generative AI for something useful in protein structure and design? And obviously it could be really useful. Here's a case where it was really useful to me, not in protein structure. I wanted a picture to show you here, and I couldn't remember the name of it. So, and you can't Google it with this query, but ChatGPT knows all about it, or DALL-E. It says, look, here's what you're thinking of. Just look at this. 36 views of Mount Fuji. In fact, here's the set of paintings. So that's the inspiration for this talk. So it could be really useful to use generative AI here. What about for protein structure? So we talked about this from my class, like what's an alpha helix, what's a beta strand. But what are these things really? Why do we do them? And why is this a form of sketching or abstraction that will help us understand what's happening? So here's a really complicated thing that Eric and I were just looking at. This is an antibody that we designed. CU and Graham and Anna designed this that actually works. And we have this really complicated structure that was solved by our collaborators. And how can we make sense of a thing like this? So let's look at this structure, which is deposited. And I'm going to look at this structure here on the left-hand corner. And I'm going to look at a little tiny piece here like this. And I'm going to pull it out so it looks like this. This is actually a protein called GP41 in HIV. And that's really complicated. So let's just look at a single helix. So what is a helix? Like we've talked about these. Is this confusing? What are these things? So here's why we draw it like this, or why it can be a good idea. So this is a view of a helix as a molecule, right? This is a thing where we have atoms, spheres filling all the spaces here. It's a pretty complicated object. If we draw it as a helix with all the side chains, just as sticks, it looks like this really complicated object here. And then down here is the basic idea. If you just look at the backbone, right, the so-called invariant part, we get this corkscrew twisty thing. And that's the motivation of Jane Richardson calling this an alpha helix. And so that you can sketch this like this using a spline. So the spline in the lower right-hand corner, this is an abstraction we use to visualize the structure, which can really be thought to be any of these things. This is spheres, this is sticks, this is just the backbone. This is just a way of sketching it to basically reduce the dimension and sketch what we have here. So maybe we don't need it for a helix, even though this thing in the upper right-hand corner with sticks, pretty darn complicated. But let's look at GP41 again as sticks. This is a pretty complicated molecule, even for a chemist or biochemist. So when we sketch that using the alpha helix, so this is the abstraction, we replace this complicated molecular thing with this spline, and we get this visualization like this. So it's a way of abstracting away and seeing what the structure looks like at a high level. And that's why we do that. And many proteins have alpha helices, almost all of them. Okay, so now we know what alpha helices are. Let's test our knowledge of alpha helices to look at some AlphaFold predictions versus ground truth. So this is, but first I want to say, what does it mean for AlphaFold to be wrong? Like, what does this mean here? And I'm going to make an analogy to DALL-E and hallucination. So first of all, let's look at this little quote from Sheldon. Are there degrees of being wrong, or is it absolute? And Stewart says, of course, it's a little wrong to say a tomato is a vegetable. It's very wrong to say it's a suspension bridge. And so I asked Dali to draw a picture of a car with some of the wheels wrong. And here's what it came up with. So here is an assembly. You could think of this like a protein assembly, right? Except the tires aren't in the right place. They're not on correctly. So even though this is kind of recognizable as a car and tires, we would say this is wrong. And of course, we have degrees of wrong here, that this is just awful. And here's another one. I talked to Eric about this, and he pointed out, look, the one on the top, that's just terrible. But the ones on the bottom don't fit either. So you can't use this for any kind of like mechanical design. And of course, in addition, it happened to put a brain in the middle here for some bizarre reason that amused my neuroscientist wife very much. So these are all things that are assembling something, physical object, in a way that's wrong, hallucinated by Dali. And so we see a similar thing happening with AlphaFold 3, which is why we should be cautious about using it for protein assemblies. Let me show you what we see. So this is a result. So, we can look at this and we can say the one on the left is worse, the one on the right is still wrong, and we could be quantitative about this if we wanted to. All right, let's look at an example. Let's test our knowledge of alpha helices now that we know how to actually visualize protein structures. Here's an example from my friend Peugeot at Duke. This isn't published yet, but will be this week. So basically, this is a protein complex that's super important in cancer that he studies. And it's a protein that has a lot of alpha helices, and it binds to a peptide that's separate, but also has an alpha helix. So basically, the blue stuff is a protein with alpha helices, and this is a separate protein or peptide, the rainbow thing here that has this long name. And what Pei found is that AlphaFold multimer, which is the precursor to AlphaFold 3, returns the structure, but the helix is completely backwards. Notice the orientation of the helix is backwards. It's colored from N terminus to C terminus in a rainbow. So it's put it in backwards the same way here we put in tires backwards, right? So AlphaFold makes the helix backwards. And by the way, this has major consequences biologically and chemically. So looking over here with other views, here we've drawn the helices just as these sort of corkscrew things, and the interfaces between these things are totally different. So if you're doing drug design, you get completely wrong results here. And also, I think Albert asked, what does the actual data look like? These aren't data, these are models. So shows the crystal structure and the AlphaFold multimer model are different. The actual data looks like this. This mesh here contains the electron density we measure by crystallography, showing where the helix really is. You can see this is really well resolved, showing it's backwards. And again, this has major implications for cancer and for structures. So now we have an idea that AlphaFold can return structures that nominally have parts that look good, but major parts of these interfaces can be completely wrong. Okay.
All right, can I ask a stupid question?
Yes.
All right, is there something that prevents it from folding in the way that AlphaFold, like, what is the energy, what is the difference in energy between, I assume it's minimizing energy?
No. This thing here is I don't know what AlphaFold's doing because it's deep learning. The crystal structure is empirical data. This is measured from actual physical measurements.
Okay, but like, what's the possibility that AlphaFold could be correct? Like, that such a structure.
There's possibility, but there's no experimental evidence showing that.
Okay. So yeah. So we have no sort of measurement.
We do have a measurement. The measurement is here. The measurement says it's different. So could it do this other thing? I mean, anything is possible, but there's no evidence for it. So it's like taking a picture of, yes, a car could do this. Someone could make this, but when you actually took a picture of the car, it looked like a normal car instead of one of these things. So at this point, we can say, I would say that AlphaFold is trained to predict the results of crystallography experiments, right? And so here we did the crystallography, Pei did the crystallography experiment, and it shows that it's backwards. So I don't think just because AlphaFold hallucinated it, we should say that there's any possibility it really does this. Like, there's certainly no evidence that it does that. Does that answer your question?
Not really. So I mean, I get what you're saying, that it's wrong. I get that. But I guess I'm just wondering if there's an energy, like if it's minimizing some kind of energy, I guess what's the difference in energy between the solution AlphaFold found and the correct solution?
So first, it's not minimizing an energy. That
Okay.
would be more like our kind of approaches, optimization approaches.
Okay.
And that could be computed, but I don't think anyone's done that. So
So we don't
I
know.
wouldn't say we don't know. So, if I used, deep learning to generate a picture of one politician pulling a gun on another one and use that for disinformation, and that picture never happened, I don't think it's quite right to say, well, we don't know that that never happened. It just didn't happen when were there with your camera, right?
Right. The analogies you're giving are not the same ones that they don't hold in what I'm trying to get at, which is let's say you would do a computation to try to minimize energy, right? The protein folding, I assuming the protein itself is trying to minimize energy, correct?
Um, so AlphaFold isn't doing any of that. The protein folding approach might minimize some kind of free energy, which is energy of binding. Yes.
Right. And so I'm just
Yeah.
asking if you ran it through that simulation, that actual computation of folding, like, would the energy of what AlphaFold produced be low?
Yeah, that's a really good question. I don't really think those. So I don't think molecular dynamic simulations of those predictions are that accurate, but one could do it. One could also run it through Osprey and ask that question. I mean, if it was, to me, it would tell you not that it was possibly correct, but it would tell you that your energy function wasn't discriminating true from false.
Right, exactly.
Yeah, but that doesn't really indict AlphaFold per se, because AlphaFold doesn't use those functions. So AlphaFold's coming at the wrong answer through a machine learning approach, and conceivably a biophysical approach that minimized energy could come up with a wrong answer from an energy minimization, physics-based approach. That could definitely happen. I guess all we're saying here is AlphaFold using machine learning, deep learning approach came up with the wrong answer. And in answer to the other question, we could also have a presentation on cases where physics-based approaches came up with the wrong answer, which is also possible.
Okay. I'll let it go now.
Yeah. And we certainly have examples of that, but okay. So the interface is wrong, these have consequences and so forth. So I want to then show you some more examples, but first a little more didactic information on protein structure. I want to tell you what a beta sheet is. So again, to answer Albert's question, this is what the data actually looks like. It's electron density around that you can fit molecules to. So electron density is a scalar field over real three-dimensional space. So it tells you where the electrons are. And based on that, we build models like this. This is called a beta sheet, the term invented by Jane Richardson here at Duke. And here are different views of a beta sheet. So one is this really complicated chemical view that has the two strands lined up with hydrogen bonds between them. So basically what Jane said, instead of this really complicated thing at the bottom, right, let's abstract away like this, where the arrow just indicates the direction of the linear chain from end to C terminus. So this blue thing is an abstraction for this thing in particular, and using that you can sketch or abstract away really complicated things. This is a particular protein that's been sketched this way. Again, this is just a visualization technique, but it's very nice way to quickly get a view of what structure looks like. So now we know what alpha helices and beta strands are in beta sheets. Beta strand is one, beta sheets are these things here. Okay. Let's go on. So my student Catherine uses AlphaFold and was really excited about AlphaFold 3. So the earlier prediction was AlphaFold multimer. This is with AlphaFold 3, the newest one. So this is a test she ran. This is an antibody we designed binding to the HIV envelope trimer. And the comparison here is to a cryo-EM structure that was solved by our collaborators and analyzed by them and by our students. This is designed by Stu and Anna and Graham and analyzed by Graham. This is a little hard to interpret the AlphaFold picture, so I visualized it. separately, but let me mention AlphaFold's pretty confident about this, but is it right? So we're comparing the AlphaFold prediction in the next slide to ground truth, which is the cryo-EM structure. And I'll come back to this slide. So again, here is the AlphaFold prediction. So I've drawn the entire HIV trimer, the protein in magenta. The antibody has two changed, cyan and green like this. And here is our structure solved experimentally. And here's a picture of Graham. And these are really different. Again, this is like the tire being on the wrong part of the car. And I've drawn the symmetry axis here, so you can see the symmetry axis and how these things line up. So basically, in the one on the right, which is correct, the antibody binds on the top. And on the one on the left, the antibody binds on the side, which is just wrong. Okay. I have more examples that I'll show now.
Okay.
Let's look at another, and I'll visualize these in PyMOL because 3D is easier, but I want to show you what I'm visualizing before I go into PyMOL. So here's another one, another antibody we designed. This one designed by CU. Again, AlphaFold's quite confident about it. I'll speak to that in a minute. This is its picture. Catherine ran this and gave me the files. Again, the test is against AlphaFold prediction versus ground truth, which is a cryo-EM structure that our collaborators solved and we analyzed. So this is the AlphaFold 3 prediction. The entire HIV envelope protein is in magenta here. Here's the antibody, which has two changed, green and cyan. And here's the correct structure binding again at the top, the Graham's analysis. Okay, so these are very different. It's binding on the side.
Can you remind me, is AlphaFold a deterministic algorithm, or does it have a random seed that needs to be set? So if you
it gives you a range of the AlphaFold 3 only gives you 5 predictions, and yet they are somewhat random. It ranks them in terms of confidence. For clarity, I'm just showing you the most likely one in its mind. They're all wrong in this case, but they don't have to be. Sometimes one of the 5 is wrong, and in AlphaFold 2, one of the 25 is wrong because you could get more. But yeah, it does have it has a random. Yeah.
Was it very confident that it was correct?
I have an analysis that says in some ways yes, but basically here's some of its confidence. So here it's colored the structure where it's confidence. This is very high confidence. You can see that's 90% for these regions here. And here's the expected position error, which is pretty small. I'm going to come back to analyze this in a minute because there is some hope there. But let me just analyze the structures and we'll come back to that analysis. So partial answer for you, Cynthia, is that since this is mostly blue, it's very confident. I have a more detailed analysis that comes out somewhat more favorably for AlphaFold, but I'll do that at the end. So again, this is the comparison, and these are the symmetry axis. Basically, AlphaFold predictions bind on the side, all of them. Correct one is binds at the top. Now, these are hard to see from static structures, so I want to show you some PyMOL visualizations, and I also want to give Eric all these pictures so he can all these scripts so he can do the same thing. So I'm going to do the same thing, but just try to turn these into scripts here. Again, here's the correct structure visualized in PyMOL. And here's the AlphaFold prediction visualized in PyMOL. We can look at these guys. All right, and get that to rock. And let's make that big. And make that big. We can go back and forth. So this is our structure in 3D with rocking back and forth. You can see it. There's a little more going on here, Cynthia, is that instead of making this whole thing magenta, I've shown all 6 chains. This is a 3-fold symmetry of 2-fold symmetries. But you can see, you can turn this around and you can see what this guy looks like. You can compare it to the AlphaFold structure, which is here. Again, HIV is this big blob. The antibody is in blue and green here. So we can visualize these things looking very different. And by the way, these are basically symmetric. I can draw this like this. So here you can see it's symmetric up the top. The antibody, of course, is not symmetric. It binds at the top. And I wrote some Python scripts here that let PyMOL do this really quickly, as you can see. Let's look at one more. and I'll then do some analysis. So here's another one. We have, in terms of data, I'll just show you a few of these. We have, I have 3 detailed examples of this. I'll only show you 2 of them in this meeting because we don't have infinite time. And I have some others that I can show too. So we have many examples of it making these kind of mistakes. Let's put these up here too. I just ran my script to do this. Rock. Oops, did not work. That's weird. There we go.
Question, did AlphaFold 3, did it train on affinities at all?
No affinities.
And it had some sort of, it had some sort of cutoff, or was it just anything that was in PDB bind?
It didn't use PDB bind at all.
It
has
doesn't.
nothing to do with binding or affinities. It's just structure prediction.
Huh? How did they get?
They trained on structures. By the way, one of the interesting things. Let me just do this analysis of this one first. This is the other one. So I want to show you the correct structure again in 3D. Again, this is symmetric if we tip it up, totally symmetric like this. This is the correct structure. The antibody binds at the top, like this. This is, PGT 145, modified by our lab to neutralize better. And this is the AlphaFold prediction, which you can see AlphaFold binding on the side. So this is completely wrong. Okay. Okay, now let me answer. Any questions about the structure visualizations? Okay, I want to now answer your question, Albert. Let me do this. Turn off the rocking and turn off the rocking. Here we go. So in answer to your questions here, I mean, one has to read the AlphaFold papers to figure out what they are doing. Let's see.
I think when I read it, how they do it is they take in PDB files, and then when they do their generations, they just train on distance between the generated thing and the actual PDB file.
Yeah, they train on structures and multiple sequential alignment. So
yeah.
So Albert, what AlphaFold 3 and AlphaFold 2 are learning is they represent structures as a distance matrix. And what they're doing is they are representing this as, they're learning a distribution, a PDF over distance matrices, and they train on structures and multiple sequence alignments. So, and I actually am asking it here to predict structures, not predict binding affinity, right? So it'd be kind of unfair to have it predict binding affinity. Does that answer your question, Albert?
Yeah, I guess I'm just wondering, how are they positioning the two, or how do they position chains even together if they're
training on complexes? They train on complexes as well as single structures.
Okay, got it. So in a sense, they're using the training from complexation.
Sure.
Probably to help inform intermolecular binding to ligands as well.
Probably, yeah. But let me mention then one interesting thing. These are just the first things we tried it on. Our structures are actually in the PDB. It could have looked these up, but it didn't. So it hasn't. They maybe didn't have time to retrain it when they released AlphaFold 3. Now, Cynthia, did you have any questions?
Nope, I'm just following along as best I can.
That's great, and I can explain anything in more detail. Let me mention a couple of other things. So, we don't have statistics for all of AlphaFold 3, but AlphaFold latest, which was the Halloween release of AlphaFold 3, said that they had increased accuracy for antibody antigen predictions from 23% in AlphaFold multimer up to 51% accuracy in AlphaFold latest, which is AlphaFold 3. That's a QDoc score of less than 0.23, 0.23, 0.23, 0.23, 0.23, 0.23, 0.23, 0.23, 0.23, 0.23, 0.23, 0.23, 0.23, 0.23, 0.23, 0.23, 0.23, 0.23, 0.23, 0.23, 0.23, 0.23, 0.23, 0.23, 0.23, 0.23, 0.23, 0.23, 0.23, 0.23, 0.23, 0.23, 0.23, 0.23, 0.23, 0.23, 0.23, 0.23, 0.23, 0.23, 0.23, 0.23, 0.23, 0.23, 0.23, 0.23, 0.23, 0.23, 0.23, 0.23, 0.23, 0.23, 0.23, 0.23, 0.23, 0.23, 0.23, 0.23, 0.23, 0.23, 0.23, 0.23, 0.23, 0.23, 0.23, 0.23, 0.23, 0.23, 0.23, 0.23, 0.23, 0.23, 0.23, 0.23, 0.23, 0.23, 0.23, it's a bioinformatic measure. So that, they're claiming an increase of accuracy, but didn't do too well on these things. All right, so we looked at some PyMOL visualizations. Eric, I'll send you all my scripts and you can visualize proteins yourself. Let me go back to Cynthia's very good question. How confident is AlphaFold of these things? So I want to tell you that when Catherine showed me her results, she's like, I wanted to use this. I'm disappointed in the accuracy. And she's a structural biologist. And so what's really going on here? So I went over these results with Alan, who's a postdoc in my lab, and he observed the following. Look at this matrix here. This is a matrix. So the axes are the number of the residue, and everything is numbered from 1 to 2300. That's a lot of residues, positions. So this is the expected position error for a pair of residues. And so what you see here is this is the little antibody up in the left-hand corner, and this is the protein. So it says it's very confident about the individual components, right? It's super about the antibody by itself and the HAV trimer by itself, but it has no confidence like 30 angstrom, which is huge in accuracy. So Alan pointed out that AlphaFold knows that it's not confident. It's telling you here that it has confidence in the individual pieces, but it has no confidence in its ability to put them together. So to put them in color codes here, it has confidence in this piece, which actually it shouldn't because it's not that good. And it has confidence in the purple piece, which is reasonable. It's actually reasonably close, but it has no confidence on the relative error of distances between the magenta thing and the green or cyan things. That's what this is telling us. So actually in this figure, if we analyze it, it's telling us that it's not confident, even though it shows us a structure. And here's the other one, same thing. This one here, what's on the side, it's telling us, remember, this is just, we've numbered this arbitrarily from 1 to 2300, where that's the numbering of the antibody and the protein. This big thing here is the HIV trimer, the big magenta thing. This is the antibody. So it's confident in the pretty confident in the errors in the antibody structure, pretty confident in the errors, pretty confident that it's low error component for the HIV structure. But again, as Alan pointed out, it's totally unconfident about how it's putting these things together. Does this make sense to you all?
Yeah.
Okay. So in summary, I suggest that to understand this, we should visualize the protein structure and protein interactions. And to get us started on that mission, I explained some ways that people abstract away to sketch these things using alpha helices and beta sheets. We've shown that AlphaFold 3 is wrong on many PPIs. And by the way, I have many more examples. So if you want to see more, I'm happy to prepare them, but we have them on other antibodies and other systems. However, the glimmer in the gloom, and that's what this title, A Glimmer in the Gloom, is that AlphaFold 3 seems to know that it may be wrong. So the expected error for interactions between the antibody and the target protein is very high. In this case here, 30 angstroms or more, right? So that's really, really big. So these distances, this is like 2 or 3 angstroms here. That's really large positional or orientational error. So AlphaFold seems to know that it may be wrong, but it's happy to give you a structure anyhow. But I think that at this point, using it in our pipeline is probably not going to be very accurate. But it might improve. So finally, let me mention this. Alan did some of the analysis here of the confidences. Catherine did the AlphaFold runs and the empirical structures, which are ground truth. These were done by our collaborators at NIH, particularly Jay and Peter. And then the designs were done by CU and Anna, analyzed by Graham. So finally, that's all I had. So I hope that was useful, and I'm happy to go into any more detail or answer any questions.
Yeah, thanks, Bruce. That's great. Just throwing out a couple ideas or thoughts. So if is there a way to, is there an opportunity where we might be thinking about different types of binding interactions, like you mentioned, this periodic table of binding. And basically what AlphaFold 3 is doing is learning kind of protein complexes, probably a lot of multimeric type structures, which could have substantially different KDs, surface areas of interaction, compared to something like a ligand to a protein, like a small ligand to a protein or a peptide to a protein. So this suggests that potentially there's like different flavors of binding interactions that need to be learned, which could be involving KD or surface area. And presumably, I'm guessing AlphaFold 3 at some AlphaFold at some point they'll go after training on PDB bind as well, right?
I have no indication they'll do that. They might. I suspect they won't because the data is pretty noisy, but who knows?
Okay.
I don't know if they're interested. I mean, a lot of people are. Let's say a lot of people in the community are interested in this. Interested in predicting binding affinities, but AlphaFold at the moment is focused on structures. But we're really just reading tea leaves. We don't know what they'll do. They're certainly capable of doing that. But no, I don't think it's necessarily the natural next step for them, just because I don't know what their interests are.
Yeah.
But I do know that AlphaFold 3 is proprietary, and you can't get the source code, and they only let you do 5 predictions, and they only let you look at protein-protein interactions or a specific number of ligands, like in their web version, because they want to use the proprietary version in a commercial venture that's pharmaceutically oriented. So they have a lot of interest in those areas, but it's not clear we get to see them.
So I'm not suggesting we do this at all. Just I'm just trying to understand what's going on here. So if one were to like run AlphaFold 3 on different types of binding interactions, and then look at which ones AlphaFold 3 is confident on and which ones that it is not confident on, that could tell us something about, what types of binding interactions it's able to predict.
Yeah, I mean, I think that they've tried to do that in their Halloween report. So basically multimers are anything with more than one protein, and they broke those down into protein nucleic acid, protein small molecule, protein where the protein is an antibody, and they have different categories for those things. So basically they try to give you accuracies for different categories, or one could run them. I mean, in their case, I think it's useful because they then try to improve those things, right?
Yeah.
But in general, the performance of the antibodies is poor for these methods. So at the moment, but it could get better. So I think that some of that data is already out there as statistics over large sets of things. That report is worth looking at. But this is just an investigation of what happened when we ran it on. Well, we ran it on about 10 different things, and I showed you 3, and plus Peugeots. So all it means, well, I was joking with a colleague in biochemistry, and he said, "Oh yeah, they have that in the code. When it's your proteins, it is guaranteed to give a poor answer." Joke, right? So yes, the first 10 things we ran it on from our lab, it did poorly on. But of course, that isn't a statistical assessment. It does say how well it should. I think this does represent how well it currently does on antibodies. I really do think it does that, because that's known. As for the other things, they're just syllogisms that we managed to catch it in.